All right, our group is under Dr. McMahon, and we're MLA using diagnosing skin cancers using convolutional neural networks. Arush and I will be presenting today, but we also have a lot of members in the group. We have Arush, Anav, Aryan, Ayan, Ishan, me, and Pranav. Okay, first we'll have 4 different sections of a presentation: background info, research, and the methods we are using, current results, and our future goals, future ideas. First up is background information. What is our project? Our project is trying to use convolutional neural networks, machine learning, to determine whether or not there is a skin cancer for an image. The first part of the research was to be able to get a data set to work with, and we use images of cancer skin lesions as the data, and for a control, pick images of skin that do not have cancer. While some of the images that go into the quote unquote cancerous section, a lot of them are categorized as melancholic nevi, which is non-cancerous most of the time. So next up, we had to clean the data because many of the images had null values. And second part of the data, load and resize images because many of the images were of different sizes. Some were, for example, 35 by 35 pixels. Others were 1,000 by 1,000. So the resolution would be different. We had to resize those to get a clean result. And next up, we created graphs before we created the CNN model because we wanted to see if any of these factors would affect our results. We created graphs for age distribution, localization of cancer, so where the cancer was on the body, and male and female distribution. We'll show those graphs in this presentation. Okay. Third part of our research, we wanted to figure out how we wanted to split the data. We decided on a 75% training and 25% testing ratio. And then we wanted to normalize the data to prevent overfitting. And this is because, again, like I said before, melancholic nevi was in most of our data, and it was about 70 something percent of it. So it will show a graph of it, but it's much higher than some of the other cancers that weren't as often showed in the data set. And the third part, well, part of the third part was to actually build a model, and Arush will show some of the challenges that we ran into that prevented us from getting to that step quicker. Last part of the research is to analyze the graphs after we build the model and finish it completely, making sure the accuracy is good. We want to double check if there are any major discrepancies and other factors outside of what we want it to affect results. For example, if the resolution is affecting what the results are giving us, then we want to go back to our model, fix that. And then lastly, run histogram tests, other analysis to make sure the model is functioning as necessary. We really want to make sure that the results are based on the actual skin cancer itself, not some outside factor that is contributing to this. Okay, main objectives for this project is one, because cancer is such a dangerous thing, we want to make sure that we can get a high accuracy. Our goal is 90% because current models for these types of cancers are around 80%, and we hope to improve upon that so that the industry, the medical professionals can use this as a guideline. Next, we want to be able to correlate factors and analyze the graphs, find out what factors are making the results skewed. And lastly, again, 90% accuracy, but this time for each cancer type. When we finish the model, we hope to single out each type of skin cancer and then produce a model specifically for that cancer and achieve 90% accuracy on those as well. And I'll pass it on to Arush.
Yeah, so let's talk about our research next. So moving on to the data collection, we have three main big data sets. The first is our skin cancer image set called the HAM 10,000 dataset. This data set has about 10,000 or so images of a bunch of different pigmented skin lesions, cancers, etc. And it's what we're actually analyzing in this experiment. The second is the skin cancer CSV. This includes metadata. Oh, sorry about that. I will put my camera on. Um, I didn't notice that I had it off. I'm so sorry about that. The second is the skin cancer CSV. The CSV contains metadata about each image in the HAM 10,000. Metadata is just extra information about each of the individual images. So for example, let's say I have a image of a melanchotic nevi cancer. Then the metadata in the CSV would tell me, okay, the person who this image comes from is 55 years old, they are a male, they have this cancer located on their upper back. And finally, we have the SFA normal image set, which is a variety of images of normal skin images. This is our control, and although we haven't integrated it into our model yet, once we get a final finished product, we hope to integrate it in as well so that we can get a more richer model. So let's move on to a couple of data distribution graphs, starting off with localization. So this graph shows the localization of different skin cancers on different parts of the body, and we used the metadata in the CSV to find this info. So as you can see, there are a lot of cancers and lesions located on the back, lower extremities, trunk, upper extremities, and generally the torso area. However, there aren't as many located on the scalp, neck, ear, or hand. Now, this does pose a small problem because when you have more data for a certain location on the body, it usually means that that location is going to give a lot more accurate results. So let's say that finding out whether or not something is a skin cancer or what type of skin cancer on the back will have about a 90% accuracy rate. But on the scalp, since it has so little data, it might only be a 47% accuracy rate. To fix this, what we plan to do is once we launch our model, we plan to run tests on each of the major localizations in the data distribution dataset, see what the accuracies are, and see, okay, well, is this something that we can actually fix? And if it is, we'll try and either add more data or move data. Or if it's something we can't fix, then we just know that we have to mention in our paper that the model isn't as accurate for, let's say, the foot or the scalp. Next, we have the data distribution of our skin cancer types. So there's a dictionary here that shows you all of the different types of cancers. Now, as you'll immediately notice, melanchotic nevi, or the NV, makes up an overwhelming amount of the skin cancer dataset. Now, this is a slight problem because it can create something known as overfitting. Overfitting is when you have too much of one type of data. So instead of actually analyzing all the different types of data and trying to diagnose the skin cancers, the model will only be able to diagnose melanchotic nevi. And obviously, our model is meant to diagnose a wide variety of skin cancers. So obviously, we want it to be able to have a good accuracy for the other ones as well. Similar to the localization, after we're done running the model, we plan on running separate models for each of these individual types of cancers and adjusting the amount of data in there so that we get a higher accuracy. We are also taking other steps to combat overfit. Right before the model enters its accuracy function, aka what percentage of the images it can actually detect that are certain types of cancers, before it actually does that, it creates a bunch of artificial images and it also deletes a bunch of images. And this forces the computer to actually learn instead of just mapping things out together. We also have the male to female ratio. So, as you can see by the numbers, there are about 5,400 males in the study and 4,550 females in the study. Now, Although a disparity of about 900 seems like a lot, in the grand scheme of things, it really isn't. And at most, it'll likely lead to only a 2% difference in accuracy rates between the two sexes. So we decided to not majorly consider it after we're done with the model. So now let's explain how a convolutional neural network, aka the very basis of our model, actually works. So a convolutional neural network, the neural network itself, consists of an input, an output, and hidden layers. So we can see how CNN works in this image. First, the input image, the image from our dataset, is divided into a lot of different pixels in sort of this grid. From there, it's put through a ton of filters to try and find trends in the image. It's usually a matrix that represents a certain feature. So for images, this might be an edge or a corner. It goes through 3D 3 by 3 grids within this input image and creates an output feature map. Some feature maps can be grouped together and put through another filter to create a second feature map that consists of a lot of different features. So think of it like this. If I have an image of a dog, my first feature map might try and look for the shape of the ears. Then it might put it through another feature map. Okay, what is the shape of the overall animal? Then it might put it through another feature map. Okay, well, is this now flared? And it's going to continue putting it through all of these different filters and feature maps until you finally have enough where you can solidly tell that yes, this is a dog. Now, we're doing something really similar here with skin cancers. We're essentially taking the image and running a bunch of filters by it to find trends in the image to try and see what differentiates one type of cancer from another. Now, the reason that we chose to use a convolutional neural network instead of individually checking the values of each pixel is it lets us find the object with a lot less storage, since it analyzes trends as a whole and doesn't try and look at a bunch of information and then create a trend. And using this, we're able to make our model a lot more efficient. So instead, the model can run in a matter of seconds instead of a matter of hours. So I'm going to show you some of the images from our image set here. But fair warning, because of the gruesome nature of skin cancers and lesions, they are a bit um, squeamish. So if you don't react that well to blood or oozing or that sort of stuff, we recommend that you look away, and I'll let you know when we are done with that slide. So these are some example pictures. On the top is our most common type of skin lesion, melanchotic nevi, and on the bottom is another one of our less common skin lesions, melanoma. So as you can see, there's usually some sort of patch that has either some sort of mole or oozing on it. And for melanoma, it's usually pretty similar, with it usually developing some non-common skin tone compared to the normal skin tone. So to build our CNN, we used instead of using an already trained and pre-existing model, we built our own. This is because CNNs have a lot of different layers and filters that it goes to try and find those trends and patterns. So we decided to create our own so that way we would have full control over the different trends that we wanted to detect. So for us, these different filters were starting off with the computational filters, the convolutional layer, which looks at two neighboring pixels and picks the minimum value, which basically just allows us to reduce the computational cost by half. Because instead of looking at each individual pixel, I'm now taking these two pixels and combining them into one, right? And then I have the pooling layer, which reduces the spatial size of the input image as a whole. This is combined with the convolutional layer, and both of these layers combined are meant to reduce the computational cost, so it doesn't take an hour or two to run the model. From there, we have the dropout layer, where some of the nodes in the CNN are randomly ignored, and this forces the network to learn features in a distributed way, which prevents overfitting. Next, we have the flatten layer, where it takes all of the feature maps that we've created and puts them into one final 1D vector, which is what allows us to actually get that final accuracy. Finally, we have the dense layer, where it classifies the image into its specific type of skin cancer based on the output and the feature map given from the convolutional layers. Additionally, since we've last presented at our at Colloquia, we have implemented a NumPy array into our code. Now, most CNNs will directly analyze the JPEG file, or the actual image itself. However, we decided to take the approach of turning our images into NumPy arrays. This is one, so it takes a lot less processing power since it no longer has to find trends such as oozing, skin tone, etc., but instead it can now look for trends in the pixels, making it easier for the computer to analyze. Additionally, because it's looking at trends in the raw numbers and raw data instead of trying to create trends by finding edges, corners, circles, etc., this leads to an overall higher accuracy for the model. Now, for those of you who don't know anything about computer science, I'm sure you're confused. What is a NumPy array and what is an array? Well, an array is a list of numbers, and in our case, we use RGB arrays. Each RGB array represents one pixel, the red, green, and blue of that pixel, showing the colors of it. And together, a NumPy array is basically just a bunch of these arrays put together into the sort of 3D model that allows us to actually see what the image would look like by showing each individual pixel. It's a lot easier for the computer to analyze trends in numbers rather than trends in images, which is why we chose this so that we can get that desired 90% accuracy. So here are some of the challenges that we faced. So we ran into 3 key challenges since we've last spoken at our last colloquium. So first was normalizing the data to prevent overfitting. So the way we did this was we artificially created and removed a bunch of images. However, this led to problems because instead of actually creating and removing new images each time the model was made to a copy of the data set, it instead did it to the base data set, which is obviously really bad because you want your data set to stay consistent so you can actually get consistent results. If you're constantly changing up your data, your base data at that too, it doesn't fare well for the accuracy. But eventually we got around it. Another issue we had was running Keras and TensorFlow on the ASDRP server cluster. Keras and TensorFlow are 2 machine learning libraries that allow us to run a bunch of neat functions that can actually analyze all those images. So, for example, TensorFlow actually has all those convolutional layers that allow us to build a model. Eventually, we decided to move all our code over to VS Code so that we can run the model locally, which would allow us to more easily run these libraries like Keras and TensorFlow, since they require a lot less setup in the long run. And finally, our biggest and most concurrent problem is standardizing the NumPy array into tensor. Unfortunately, convolutional neural networks don't accept a NumPy array. You have to convert whatever it is you're analyzing into something called a tensor, which is required to actually get the accuracy function of the model. And currently, we haven't found a solution to this, but once we do, we'll have our first model up and running, and hopefully we'll get the 90% accuracy we've been desiring.
Okay, lastly, we have our future goals. We have 3 future plans once we get our model up and running. First one is, of course, finish the actual model, and we hope to figure out the tensor problem by March 18th, which is coming up soon. And then our second goal is to, after we finish the main model, create a small model. Because melanchotic nevi takes up so much of the percentage of data, we want to create a separate model just to be able to identify melanchotic nevi. And lastly, we want to be able to use 3D images into the experiment as well, because that way we get to see the depth or the height of how big each cancer looks like. And that's going to be a separate factor that goes into our model. And we hope to use a data set that's similar to the HAM 10,000 because the HAM 10,000 was a pretty good match for us. It had a substantial amount of data for our purposes, and it is representative of the major diagnostic categories for pigmented lesions, benign or not. So whether or not it is cancer, this HAM 10,000 had both cancerous and non-cancerous images, and we hope to find, if we're going to do a 3D model, we hope to find something similar to that. And that's it for our presentation. Thank you for listening. Any questions?